Hello everyone, my name is Daniel Ford. I'm the producer and presenter of Pay-Per-View, a podcast analysing news and current events and connecting the dots the mainstream media refuses to acknowledge, never mind connect. In this video I'm going to analyse scientific papers and, through pointing out their common flawed methodology, explain why, although they all claim to have done so, none of them have actually described genuine isolation of SARS-CoV-2, the virus claimed to cause Covid-19. So what is a virus? Viruses are the smallest of all the microbes. They are said to be so small that 500 million rhinoviruses, which cause the common cold, they say, could fit onto the head of a pin. They are unique because they are only alive and able to multiply inside the cells of other living things. The cell they multiply in is called the host cell. A virus is made up of a core of genetic material, either DNA or RNA, surrounded by a protective coat called a capsid, which is made up of protein. Sometimes the capsid is surrounded by an additional spiky coat called the envelope. Viruses are capable of latching onto host cells and getting inside them. That's the official line on viruses. So, isolation of viruses. This is the most important step. If you don't isolate a virus, you can't prove it exists, you can't understand it, you can't photograph it, you can't test for it, you can't sequence it, you can't characterize it. You have to isolate the virus away from any other material. So you take a body sample, let's say for a respiratory virus it was lung fluid, or bronchoalveolar lavage as they call it in scientific papers, and you put it through a filter, and the pore size of the filter is small enough to let virus particles through, or virions as they're called, but too big for bacteria and other material that is in the sample, the lung fluid in this case. So here's a description here. Unlike bacteria, many of which can be grown on an artificial nutrient medium, viruses require a living host cell for replication. Infected host cells can be cultured and grown, and then the growth medium can be harvested as a source of virus. Let's talk about the cell culture method, which I'm going to get into in the next video. Virions in the liquid medium can be separated from the host cells by either centrifugation or filtration. Filters can physically remove anything present in the solution that is larger than virions. The viruses can then be collected in the filtrate. Now, filtration and centrifugation are the key stages. If you don't do those stages, you've not isolated the virus. So once you've got the filtered fluid, you then put that fluid through a centrifuge, and if there's any, it's basically a piece of laboratory equipment with a wheel powered by a motor. It's a box basically, and the sample material, the fluid, is spun at high speed using centrifugal force, and if there's any viral particles in there, they will form a band of material, and you can then extract them with a pipette and look at them under an electron microscope. And you know that any genetic material, anything you see, anything you take from the virus, comes from the virus. It is the virus, as that's the only thing you have. So this is an article on the Journal of the Royal College of Pathologists of Australasia, and it's interesting what it says here. Virus culture has been regarded as the reference standard of diagnostics for decades, as it allows for identification and isolation of active replicating virus. But the cell culture method is not isolation. Isolation means separating from everything else, but what they do in cell cultures is mix with a load of other things, but they call it isolation. However, more rapid and sensitive molecular technologies, typically nucleic acid amplification tests such as RT-PCR, which I'm going to get into in another video about the sequencing, are now the major routine diagnostic tests used in virology diagnostic laboratories. And it does seem that there's a possibility that we're moving to a point where viral culture won't even be used. They'll just go straight to sequencing. So, no isolation, even in a viral culture form. So this is the first article I'm going to look at. This is said to be the first, if not one of the first articles published Isolation and rapid sharing of the 2019 novel coronavirus and the first patient diagnosed with Covid-19 in Australia. And the most important section to look at is the methods. The methods section tells you what was actually done. Material from the initial nasopharyngeal swab, that's a nasal swab, was used to inoculate a virus-lab cell line. So that's a viral culture, which is not isolation. They did centrifuge, but without filtration. Centrifugation on its own is not isolation. Whole genome sequencing, detection of SARS-CoV-2 in clinical samples. An nasopharyngeal swab and sputum were positive using RT-PCR, that's the sequencing. I'll get into that in the sequencing video. 2 days after the inoculation of the virus-lab cell line, a subtle viral cytopathic effect was observed. That's what they say the virus does in the cell culture, but I'm going to get into why that conclusion is flawed in the next video. And I'm going to talk about in the next video where these images come from, which are allegedly the virus. Phylogenetic analysis, that's a sequencing discussion, and then that's the paper. So non-isolation there. And if you scroll down sometimes to the bottom, you find supplementary information, supplementary methods, where they go into the methods more, or what you will sometimes see is a link to a full text version of an article. Sometimes all you will see is something like that, just a brief description. If you click on the link to the full text, you get that, all the information. So this is the supplementary information. Virus-lab cells were grown at 37 degrees room temperature in maintenance media consisting of Earl's minimum essential medium, fetal bovine serum, glutamine, sodium pyruvate, sodium bicarbonate, and genoticin. Now you will see the same kind of material mentioned for cell cultures when you read papers, and I'm going to get into that in the next video. So, non-isolation. Now this is a key paper. This is cited a lot, this one. Identification of coronavirus isolated from a patient in Korea with Covid-19. Virus isolation. The virus was isolated from nasopharyngeal and oropharyngeal swabs, nasal and oral swabs, from putative, that means suspected, Covid-19 patients. Oropharyngeal samples were diluted with viral transfer medium containing nasopharyngeal swabs and antibiotics. They're a staple of cell cultures. Before being inoculated onto vero cells. Inoculated vero cells were cultured at 37 degrees to Becker's minimum essential medium, supplemented with fetal bovine serum and penicillin and streptomycin. Virus replication and isolation were confirmed through cytopathic effects, gene detection, and electron microscopy. Gene detection is obviously connected to the sequencing. That's the sequencing, introduction, sequencing, electron microscopy, virus isolation, and then it goes into the sequencing. Structure of what they say is the virus, full-length genome sequencing discussion, that's the paper. No isolation again. But what was interesting about this paper was, it says here in the introduction section, following the first outbreaks of unexplained pneumonia in Wuhan, China in late 2019, a new coronavirus was identified as the causative agent in January 2020. Now that's a declarative statement, that's a definitive statement there. So when you click on that link, this is the paper they've used to back up that statement, that reference. Full genome evolutionary analysis of the novel coronavirus rejects the hypothesis of emergence as a result of a recent recombination event. It's looking at the similarity between what they say is the genome of the virus in bats. Nothing to do with proving a virus. SARS-CoV-2 isolation from the first reported patients in Brazil After diagnosis in patients that returned from Italy to the Sao Paulo city in late February by RT-PCR, SARS-CoV-2 isolates were obtained in cell cultures and characterised by full genome sequencing, electron microscopy, and in vitro replication properties. That means in laboratory in a cell culture. The virus isolate was recovered from nasopharyngeal specimen propagated in vero cells, E6, that's the most common type of vero cell they use. CCL81 and H-SLAM are common as well, with clear cytopathic effects and characterised by full genome sequencing, electron microscopy, and in vitro replication properties. Materials and methods: we use Vero E6 cells for isolation and initial passages. We culture Vero E6 in minimum essential medium, fetal bovine serum, etc. Filtration, platform units, negative stain microscopy, virus growth, sequencing. RT-PCR positive. Just looking at the sequencing again. Post-infection of the cells. There's an image of what they say is the virus. Growth kinetics, virus titration. Basically, that's the methods described. Now, this is another key paper. A novel coronavirus in patients with pneumonia in China in 2019. In December 2019, a cluster of patients with pneumonia of unknown cause was linked to a seafood wholesale market in Wuhan, China. A previously unknown beta coronavirus was discovered through the use of unbiased sequencing in samples from patients with pneumonia. Human airway epithelial cells were used to isolate a novel coronavirus named 2019 nCoV, which formed a clade within the subgenus Sarbecovirus, orthocoronavirinae subfamily. Different from both MERS and SARS, 2019 nCoV is the seventh member of the family of coronaviruses that infect humans. Now, the key line in that paragraph is this: in December 2019, a cluster of patients with pneumonia of unknown cause was linked to a seafood wholesale market in Wuhan, China. Because Wuhan is notorious for toxic, polluted air. Respiratory illness in Wuhan is as familiar as seeing the sun in Jamaica. What else would you expect with such an environment? And Lombardy in Italy, northern Italy, which was said to be another major centre of this virus outbreak, is the same. So what does it say about the methods here? 4 lower respiratory tract samples, including bronchoalveolar lavage fluid, lung fluid, were collected from patients with pneumonia of unknown cause who were identified in Wuhan on December 21st, 2019, or later, and who had been present at the Huanan seafood market close to the time of their clinical presentation. 7 bronchoalveolar lavage fluid specimens were collected from patients in Beijing hospitals with pneumonia of known cause to serve as control samples. Bronchoalveolar lavage fluid samples were collected in sterile cups to which the virus transport medium was added. Samples were then centrifuged, but not filtered, to remove cellular debris. The supernatant was inoculated on human airway epithelial cells, which had been obtained from airway specimens resected from patients undergoing surgery for lung cancer and were confirmed to be special pathogens by next generation sequencing. No filter, no filtration mentioned. Microscopy sequencing, just looking at the patient. That's what they claim is the virus there. Sequencing, discussion. Again, no isolation. Isolation and characterization of severe acute respiratory syndrome coronavirus 2 in Turkey. Virus isolation. A patient was admitted to Kayseri City Training Research. The patient's nasopharyngeal sample was obtained by using a kit containing viral transport medium. Diluted sample was inoculated onto Vero E6 cells and gently agitated at 37 degrees. Consequently, DMEM, minimum essential medium, with fetal bovine serum was added, and the infected cells were monitored for the appearance of cytopathic effects. They don't mention centrifugation, they don't mention filtration. Cell lines there, synthesis PCR, like I say, as a test basically. Virus titration, analysis of Western blot, sequencing. Looking at the effect of what they say is the virus in the cell culture. Titration. Sequencing, discussion. Discussion is just basically summing up what they did and found. No isolation there. Virus isolation from the first patient with SARS-CoV-2 in Korea. This is another often cited paper. So what did they do? The patient's oropharyngeal samples were obtained by using kit viral transport medium. On day 7 for illness, we inoculated monolayers of vero cells with the samples and cultured the cells at 37 degrees and 5 degrees in 5% carbon dioxide atmosphere. Did they centrifuge? Did they filter? Doesn't look like it. That's the sequencing. Sequencing. That's it. And then isolation again. It's like Groundhog Day reading these papers. Now this is an interesting one. This was an article published by the CDC, the Centers for Disease Control and Prevention in America. Severe acute respiratory syndrome coronavirus due from patient with coronavirus disease, United States. Methods. Specimen collection, cell culture. We use vero cells for isolation and initial passage. We cultured vero cells, vero E6, CCL1H in Dobeca minimum essential medium, supplemented with heat inactivated fetal bovine serum and antibiotics. We use nasopharyngeal and oropharyngeal swab specimens for virus isolation. We then trypsinized and resuspended vero cells in DMEM containing 10% fetal bovine serum, penicillin, streptomycin, antibiotics, etc. Amphotericin, that's an antibiotic. We then grew the inoculated cultures in humidified 37 degrees incubator. Sequencing, microscopy, results, discussion. This is published by the CDC, just the same as all the others. Now, this is an interesting one. Characteristics of SARS-CoV-2 isolated from asymptomatic carriers in Tokyo. We inoculated the monolayers in vero and vero E6 cells distributed by the Osaka Institute of Public Health. By the way, the cells, they can be cells that commercial cells you can just buy to use in a laboratory. With 3 clinical specimens to isolate SARS-CoV-2, the cells were grown in Eagle's minimum essential medium, 2% FCS. That may be meant to be FBS, I don't know. At 36 degrees and 5% CO2 in a biosafety level 3 laboratory. 3 days after inoculation, cytopathic effect of one specimen was observed in the vero E6 culture but not in vero cells. Sequencing, introduction, no isolation. Detection and isolation of SARS-CoV-2 in serum, urine and stool specimens of COVID-19 patients in the Republic of Korea. Virus isolation. To isolate SARS-CoV-2 from samples that tested positive for the virus in RT-PCR analyses, the samples were mixed with penicillin, streptomycin, antibiotics. The samples were then centrifuged but not filtered, and the supernatant was used as the inoculant. Supernatant is the cell culture fluid, but the cell inoculation cells were cultured from the cell line cells in DMEM supplemented with 20% fetal bovine serum and 1% penicillin. We're incubated at 37 degrees. DMEM, fetal bovine serum, centrifuged but not filtered. Viral copy letter, ethics statement, results and discussion, sequencing, specimens, introduction, no isolation again. And I could go on all day, and it would be the same. I've read 40 papers in total, and this is on the pay-per-view website. This is a comprehensive, detailed, but hopefully cogent explanation of the methods used in modern virology and their flaws. The cell culture, the test and sequencing, I talk about it all in this monograph, available at paperview.uk. And these are all the papers I've read. The papers you've seen in this video are in this list, and here's some of my references. So I hope that's been informative. I hope that's easy to understand, and I hope you can apply that knowledge yourself and read other scientific papers. Stay tuned for the next video where I'm going to talk about viral culture. Thanks for watching.